we're going to do the easiest part of Unit 3 first, and that has to do with enzymes, because Unit 3 is really all about metabolism and two really, really important metabolic processes that go on in cells: photosynthesis and respiration, right? So in order to understand those processes, you have to understand enzymes, because enzymes actually do the bulk of the work of photosynthesis and respiration. I always say in baby bio class that even if it's not expressly stated in notes or whatever, you have to consider that enzymes are involved in every single cellular process. So even if I don't say enzymes are involved, chances are enzymes are somehow involved in every single metabolic or any kind of cellular process, but especially cellular metabolism. And by the way, I've been using this word metabolism a lot. Let me just remind you that metabolism is the sum total of all of the reactions that take place in the body of an organism. That includes anabolic reactions, which are reactions that put together biochemicals, but also catabolic or digestive reactions that take chemicals apart. And oftentimes, that really occurs in two steps, right? You digest things from the food that you eat, right? And then you siphon off the products from that and use those products to create new molecules inside of the body. So they kind of work hand in hand. But enzymes are responsible for doing a bunch of them. By the way, the cellular energy molecule fueling metabolism is ATP. We'll get into that a little bit later on. But the last phosphate group in ATP releases 7.6 to 7.8 kcals of energy, depending on which textbook you're reading. All right, so enzymes act as catalysts, and what that means is that they change the rate of the reaction, but they're not used up by the reaction. So they're going to hold on to whatever it is that you're trying to react to allow that thing to react, but they themselves are not going to become a part of that reaction, which means that enzymes are reusable, right, as part of this, because they can catalyze a reaction, let go, and then go grab something else and catalyze another reaction. Most of the time, enzymes are proteins, although RNA can catalyze its own reactions, but we don't typically think of them as being enzymes in the classical sense. An enzyme's job then as a catalyst is to lower the activation energy of a reaction. And activation energy, you'll recall from chemistry, is the energy that's required to make a reaction begin, right? Usually that's heat. In chemistry, you put your little Bunsen burner underneath it and cooked it to get it to start. Sometimes it's electricity. It really just depends. But whatever's required to get enough kinetic energy into the molecules to either get them to interact with one another or to break apart, depending on what kind of reaction you're doing. Enzymes also are specific to their substrate because they have an active site, which is the region of the enzyme that actually binds to the substrate, that actually conforms to the substrate. And that's a very, very specific thing. So just like we learned back in biochem that proteins' shape is important to their function, that's doubly true with enzymes. That enzymes have to actually fit the substrate that they're acting on. This is referred to, by the way, the fact that an enzyme can actually conform to whatever it's binding to. So if you see here, here's my substrate, here's my enzyme. It's got the basic shape, right? But then with enzymes, they actually kind of conform as the substrate binds to the enzyme temporarily. They conform to it. That's the induced fit model that you see mentioned there. This is kind of like clasping into a handshake or a lock and key mechanism. Usually that's how that's defined or described. So here's a few pictures to go along with what we're talking about. So this, if you're looking on the x-axis, you've got progress of the reaction, y-axis, free energy. Notice the black line here is the reaction without the enzyme. Okay, so it takes a whole lot more energy. You have to cook this a lot more to get it to start to react to products. When you add an enzyme, of course, this reaction goes with a lot less energy involved. By the way, this delta G, this free energy, the Gibbs free energy concept is not one that you need to worry about for AP. Just understand that we're looking at less energy being required to make the reaction go here. Okay, so don't worry about Gibbs free energy. And this little diagram demonstrates the induced fit model. If you look at the shape of this enzyme, you can see that this kind of fits these particular substrates, right? But notice that once the substrates bind into the active site, the enzyme actually clamps down on them a little bit. So the binding of the substrate to the enzyme actually improves the shape of the enzyme's active site to make it fit even better. That's what induced fit is. So here you have substrates, they're binding into an enzyme. That active site now allows us to convert these to products. Once this is over with and the products are released, the enzyme goes back to its original state and can pick up new substrates. So again, the enzyme is not consumed by this reaction. All right, now there are several things, of course, that can impact the rate at which an enzyme reacts. You have denaturing, and in baby bio, we talked about pH or temperature changes, right? So we said typically high temperature and low pH. That's not always the case. You can also add high salinity to that. Salts, you'll recall from chemistry class, are a cation and an anion ionically bonded together, right? A metal and a non-metal bonded together ionically. And those enzymes or those ions in solution can actually interfere with the tertiary and quaternary structure of an enzyme. So they can actually interfere with polar interactions or ionic interactions that are present in the tertiary and quaternary structure of the enzyme and actually pull it apart and make it change its shape. Once enzymes denature, they don't typically renature. They can, but not typically. We typically think of denaturing of a protein, like if you think about gelatin, right? When you heat it, what happens is this coiled up nice little protein complex just kind of falls apart and becomes a long string and it becomes liquid. And then as it cools back down, the protein can actually renature. And some proteins can, but most of the time our enzymes do not. You can also have cofactors or coenzymes, and I always think of these as an enzyme's little buddy, that is an attachment to the enzyme's active site that kind of scaffolds the active site into a specific shape. So it kind of helps hold the enzyme into the right position so that the active site remains in its proper shape. A lot of vitamins that you take act as coenzymes. So just a little buddy to help the enzyme keep its shape. You also have some other things that could happen with enzymes. You can get inhibition with enzymes, where something is preventing the enzyme to work. You're not denaturing the enzyme, but you're actually preventing it to work. The most common and easiest to understand is competitive inhibition. And this is where you have something that is close to the shape of the substrate, but it's not exactly the shape of the substrate. And it's capable of binding into the active site. And once it binds in, the enzyme locks down on it. But because nothing ever gets converted to products because it's not actually the correct substrate, the enzyme doesn't let go. So now that active site is blocked, and now that enzyme is done. It's not reacting with anything. You can also have non-competitive inhibition. Sometimes what happens with enzymes, and I think I have a picture here. Yeah, here's a non-competitive inhibitor. It's easier to understand with a picture. You've got this nice enzyme here with its nice shape, and there's a site that's not the active site. It's at the back side of the enzyme. And when a non-competitive inhibitor binds into this site, it changes the shape of the active site so that now the active site will not conform to the substrate. So here's competitive inhibition, right? This is blocking the active site. This is non-competitive inhibition, where binding someplace else on the enzyme changes the shape of the active site. Okay, so that's non-competitive. A lot of toxins and poisons will do this. So those things will actually bind in on another place in the enzyme and make the enzyme kind of change its shape. Now, non-competitive inhibition is one example of an allosteric interaction. Allosteric literally means other site, I think. The allo definitely means other. In other words, with non-competitive inhibition, what you have is kind of a permanent change to the enzyme. But you can actually have allosteric regulators that will bind to another place on the enzyme, similar to non-competitive inhibition. But those inhibitors can actually, when they can shut down the active site just like a non-competitive inhibitor, but they also can come off. And when they come off, the active site goes back to its normal shape and the enzyme can work again. You can also get allosteric activation, where you have an enzyme that's kind of got a wonky shape, and then the activator binds to this other site on the other side, and it automatically changes the shape of the enzyme. So now the enzyme's active site is exactly what it should be. So allosteric regulation can be inhibitive, but it's usually temporarily inhibitive. And then it can also be a situation where it's activating. And this makes sense because there are a lot of really super active enzymes inside of cells, and we don't need those enzymes going around reacting all the time. We don't need them to constantly make products for us. And sometimes they're really strong digestive enzymes that would break down the internal components of cells. You can't have that, right? So using these allosteric regulators, enzymes like that can be shut down temporarily until they're actually needed. You can also have something called cooperativity, where enzymes have multiple active sites. So the induced fit at one active site actually stabilizes other active sites on the enzyme. And I think I've got a picture of that. Yeah, here's cooperativity, right? So the substrate binds into this little active site right here, and immediately all the other active sites get into their proper shape. So they're capable there. Here is allosteric activation, right? So here's our enzyme. We put the activator in and it stabilizes the active sites, right? An inhibitor would bind in and keep the active sites from stabilizing. Okay, but again, that's at a place other than one of these four active sites. All right, the last kind of concept here with enzymes deals with metabolic pathways. And what you're going to see a lot of times is you get these reactions where A reacts with B and that produces product C. And product C will actually bind with some enzyme to create product D. Product D plus product E in the presence of an enzyme will make product F, and so on, right? So you get step-by-step reactions, and enzymes are typically involved in all of these steps. You can actually have what we call feedback inhibition occurring, where the production of an end product, say product H, right, goes up and inhibits the enzyme that makes product C. So you shut that pathway down at the beginning and acts as so you've got this inhibitor that shuts it down. So here's a great example. Threonine, which is an amino acid, this is threonine, can be actually converted to isoleucine in the body when you need isoleucine. So here's the enzyme, right? Threonine deaminase, I guess is how you pronounce it. It produces an intermediate that reacts with an enzyme to produce another, and so on and so on. And the final step in this reaction is isoleucine. But once you get enough isoleucine building up inside of cells, you don't need to keep making it. And so that isoleucine can bind to an allosteric site on this particular enzyme. And when it's bound, notice it inhibits the enzyme. When isoleucine is in short supply, this isoleucine gets pulled off and used, and then that activates the enzyme, which triggers this metabolic pathway to continue. Okay, so you can actually get that happening an awful lot in these metabolic pathways that involve enzymes.